Okay, welcome everyone, and thanks. I'd like to thank the organizers, especially Duncan, for asking me to come along and talk about my work. So we study voltage-gated potassium channels, and I don't need to give much of an introduction, so I'll just kind of whiz through because I have many slides. So we study in this talk, I'm going to talk about KCNQ channels. So Al just spoke about those. So we're concentrating on the neuronal KCNQ channels. There's 5 in the family, and we're looking primarily at KCNQ 2 through 5. And these, when they're activated, they limit neuronal firing. So they activate around here in the action potential, and if they're activated, they basically slow down or reduce the number of spikes in an action potential train. Loss of function, primarily loss of function. There are some gain of function mutations, but mostly the loss of function mutations in KCNQs are associated with epilepsy, and there's just some examples there. So, as Al also mentioned, Ratitobeam was a first-in-class anticonvulsant that works by opening neuronal KCNQ channels. And other groups had previously found that Ratitobeam binds to a binding site on KCNQs, and an essential element of that binding site is a tryptophan on the S5. Right here. In KCNQ3, it's W265. So to start off with, we took Rob McKinnon's KCNQ1 cryo-EM structure, and we changed the structure of this a little bit to make it look more like a neuronal human KCNQ3 channel. And then we did docking with just a simple Swiss dock technique, which is a website that anyone can use to look to see if we could find Ratitobeam docking within those channels. And we found, exactly as other people had shown experimentally, that Ratitobeam docks very close to this tryptophan. So this docking system worked. Other groups, especially Kim and colleagues, published in Nature Communications a few years ago, showed that an essential part of Ratitobeam that allows it to interact with this tryptophan is a negative electrostatic surface potential close to the carbonyl group here. And other anticonvulsants in development, such as ML213, have the same feature. So we were interested in that, and we thought the tryptophan on the S5 of KCNQs has been around for about 600 million years. It obviously didn't evolve with the purpose of binding Ratitobeam, and it's not in almost all other voltage-gated potassium channels. It's just in the neuronal KCNQs. It's in all the deuterostomes. It's in Hydra, which is kind of interesting as well. It's not in the protostomes, but it's been around for over 500 million years. So we asked the question, could this have evolved to be activated by an endogenous small molecule? So we looked at the essential chemical components of Ratitobeam, as the other people had previously found, and that negative electrostatic surface potential in particular. And we looked at endogenous small molecules, metabolites, neurotransmitters that might have that feature. And an obvious example to me was GABA. GABA has a carbonyl, has this electrostatic surface negative potential, which is in red here. And this is an inhibitory neurotransmitter, whereas glutamate, for instance, very similar in structure, but does not have the same electrostatic surface potential here. So we tried GABA. What we do in my lab mostly, the high throughput technique we use is we use 2 electrode voltage clamp, and we have postdocs that require very little sleep. So first of all, we tried expressing all the KCNQ channels in oocytes. And remember, I told you that KCNQ1, which is in the heart and epithelia, does not have this tryptophan on S5, whereas the neuronal KCNQs do. What we found was when we used tritiated GABA is that tritiated GABA binds very nicely to the neuronal KCNQs, oocytes expressing those, and does not bind to KCNQ1 expressing oocytes. We can compete out the tritiated GABA with cold GABA, and we can also calculate binding affinities. So for KCNQ3, the KD for GABA is 126 nanomolar. So this is it's kind of heretical. A voltage-gated channel should be able to bind a neurotransmitter. I believe this is the first example. So what happens functionally, that's important. Well, if we looked at all the KCNQ1s in turn, GABA basically, even though it binds to KCNQ2 through 5, it only activates KCNQ3 and KCNQ5. And you can see here the binding is highly efficacious in the case of KCNQ3. For KCNQ5, the potency is very high in the low nanomolar range, but the efficacy is much lower. So here's an example, as Al showed you for Ratitobeam. What GABA does is it left shifts or negative shifts the voltage dependence of activation, so they can activate a much more negative potentials, which we be predicted to be inhibitory in the nervous system. As a control, we tried many, many controls, and I don't have time to go through them today, but you can, if you're interested, you can read the paper came out last year. But for instance, glutamate, which has a very similar structure but does not have the same charge, is ineffective even up to 10 millimolar. It has no effect on KCNQ2-3 channels. So this is the primary correlate of the M current, whereas GABA nicely activates. The EC50 for activation we calculated around 1 micromolar. The binding is around in the hundreds of nanomolar. And this is quite interesting because tonic GABA in the brain is around 160 nanomolar. So we think that KCNQ channels have evolved to sense tonic levels of GABA and actually other metabolites and neurotransmitters as well. And rather than maybe being involved in rapid synaptic transmission, they might be able to sense tonic levels and adjust excitability on a kind of a longer time scale. Going back to the tryptophan, when we mutate the tryptophan, that our docking predicted was where GABA would bind, we get rid of the effect of GABA. So it looks like that tryptophan is important. And then when we mutate the tryptophan and look at tritiate GABA binding, we also greatly reduce the GABA binding. All right, so far so good. So now I'm going to talk about another series of projects that we work on in the lab. So around about the same time that we're figuring out that small molecules such as GABA could bind to the KCNQ channels, we started looking at traditional medicines as well. And one of the things we were very interested in is an African shrub called Malatus oppositifolia, which has been used for centuries as quite an effective anticonvulsant. It's in the scientific literature as well, but the mechanism wasn't known. So we screened several of the compounds that are known to be in the Malatus plant, and the most obvious one, which had previously been found to activate KCNQ1, which was malatoxin, or also called rotlerin. We find it very effectively activates neuronal KCNQ channels, especially KCNQ2 and KCNQ5. You can see the dose response here. So when we mutated and docked and to look for the residue that was important for malatoxin binding to KCNQs, the results predicted that the tryptophan that I talked about before was kind of in the vicinity, but actually this arginine, which is positioned between the S45 linker and S4, was more important. When we mutated this arginine in KCNQ2-3 channels, we completely get rid of the effect of malatoxin. So we think that this arginine is highly important. And this is just looking at the change in voltage dependence of activation. So we took malatoxin, we injected it into mice in a pentylene tetrazole anticonvulsant assay, and unfortunately, it wasn't really that effective. So we went back and thought maybe there's something else in these plants which is more important, or maybe acting in combination. And we went through many different compounds, and one of them, which is very interesting, isovaleric acid. So it actually looks quite a lot like GABA. It's got this nice electrostatic negative surface potential around a carbonyl group again. And as many as 2 million Americans take isovaleric acid every week because they take valerian root to get to sleep or reduce anxiety, and isovaleric acid is in valerian root. It's also in Malatus, and it's also, reputedly, the Pied Piper didn't really attract the rats with music, but rats love valerian root. So if you put it in your pockets, they'll follow you around, and the actual legend is that he was using valerian root. Anyway, I'm off topic now. So isovaleric acid was predicted to bind, in the docking was predicted to bind much more closely to the tryptophan, the S5 tryptophan, than we saw for malatoxin. It activates KCNQ2 channels especially quite nicely. And when we mutate the tryptophan here and look at isovaleric acid, we get rid of completely of the effect of isovaleric acid on KCNQ2-3 channels. Whereas if you look at malatoxin, I showed you that the arginine was very important in the voltage sensor. If we mutate the tryptophan and look at malatoxin, there's only a slight shift. So this predicted that isovaleric acid is much more tightly bound to that tryptophan than malatoxin, which docks, if you do docking studies, they're predicted they could actually dock together in the binding pocket. So we thought, okay, let's combine those two compounds from the plant. And sure enough, when we combine them, if we look at the effects of current increase at minus 60 millivolts on KCNQ2-3 channels, we can see small effects of either compound together, but the currents increase 25-fold when you add them together. We also took a range, a cocktail as we call it, of many of the other compounds in that plant, and they don't really increase it more than just these two together. When we look at survival in the PTC assay, which is quite a harsh chemoconvulsant, again, only the combination of malatoxin and isovaleric acid is effective in protecting the mice against seizures. So we think that this is the molecular mechanism for this African shrub that's been used for a long time. Just quickly, we have an approach now in my lab where we leverage the heteromeric composition of KCNQ2/3 channels, so we combine compounds that like to activate via KCNQ2 with those that activate via Q3. Ratitobeam prefers Q3, isovaleric acid and MTX prefer Q2. So when we do combinations, here's Ratitobeam. When we add it with isovaleric acid, we get more of an effect. When we add it with MTX, we get even opening at very negative potentials. When we put all three of those compounds together, we can lock the channel open completely, so it's open all the time. So you get around an 80-fold increase in current, and you can see that the voltage dependence is now completely gone. It's locked open. You wouldn't want to do this to a patient necessarily, but we know that patients with Ratitobeam have side effects, including they turn blue. So if you can use the plant compounds in combination with low concentrations of Ratitobeam, we show that you can greatly increase the efficacy of Ratitobeam. So one approach could be in the future to get around the toxicity of Ratitobeam is to combine it with certain plant compounds to reduce the dose that you need to use. All right, very quickly, I'm going to talk about some other stuff in my lab. So many of you won't have heard of Mellitus, but you've all heard of Coriander, or Cilantro as it's called in the United States. And we did the same approach with Cilantro. Cilantro has been used as a folk anticonvulsant in the Caribbean and South America and Latin America for again, many centuries. And we have the mechanism. I don't have the details, time to go through the details today, but this was published in FASEB Journal this year. We found there's one compound called E2-dodecanol in Cilantro, which really effectively opens KCNQ2/3 channels, and we have evidence to show that this is the molecular basis for the anticonvulsant action of Cilantro. So think about that next time you're eating tacos. So the last part of my talk is just got published. We're really excited about this. So KCNQ channels, I've been talking about them in the brain. KCNQ5 is in the brain. It's linked to epileptic encephalopathy, but it's also in the blood vessels. And if you open KCNQ5, it's in the smooth muscle surrounding blood vessels. You can actually vasodilate. So it's a possible target for blood pressure, high blood pressure. So what we did, we took this approach where we said, let's look at all the plants or 10 different plants that have been used traditionally to lower blood pressure. So many of these plants have been used for thousands of years as food or medicine, and more recently people have realized, oh, these actually do lower blood pressure. There's interesting evidence in the fossil record that even early hominins such as Homo erectus were actually using medicinal plants. I don't have time to go into that today. What we did, in red we took 10 different plants that have been shown, at least in preclinically, to lower blood pressure, and in blue we have 5 controls. So these are either plants that don't lower blood pressure, or they're plants such as parsley that lower blood pressure but as a diuretic, so not expected to open Q5. And we do a methanol extraction. We take a 1% dilution of that extract, and we look at the effects. This is just the sum total, and it was just, I'll show you some in detail, but it was incredibly striking. All 10 of the hypertensive plants, things like thyme, oregano, marjoram, basil, lavender, chamomile, fennel seed, cushen, and Vietnamese sephora root, and ginger, they all activate KCNQ5 channels, every single one. None of the 5 controls, which are mint, tarragon, curly parsley, Italian parsley, and wheatgrass, none of them activate Q5. So it was completely stunning. We kept doing controls, more controls, to try and rule out, but it's black and white. All the hypertensive herbs activate Q5, and none of the non-hypotensives do. Just to show you a detail of 3 of the positives here, lavender is incredibly effective at opening KCNQ5 channels. Same for fennel seed and cushen. Slightly less effective at shifting the voltage dependence, but good at activating at more positive potentials. And again, here's a couple of controls. They don't do anything. Look at the membrane potential. It's greatly shifted by these, whereas the negative controls don't do anything. The statistics, each one of these points is a different plant. So basically in red, the hypotensive herbs activate Q5. They don't activate Q2/3, and the non-hypotensives don't do anything to either. And if you look at the shifts in membrane potential, KCNQ5 expressing oocytes, hypotensive herbs shift all the plants shift Q5 expressing oocyte membrane potential. None of them activate Q2, Q3, and none of the hypotensives activate either. So it's really black and white. We went to one of these plants, cushen, which is a traditional Chinese medicine, Sephora flavescens, and we looked at some of the kind of key molecules that had already been found in that plant, and we found one alloperine that activates KCNQ5 really nicely, matrine and oxymatrine. have a tiny effect, which may add to the sum total of the plant, but by no means would you say they were activating on their own. But Q5 really nicely activates. That's 100 micromolar, I think it's allopurin, 100 micromolar, and also shifts the voltage dependence. In blood vessels, Tom Jeps in University of Copenhagen, he does an experiment where he uses myography. He pre-contracts the blood vessels, and then he looks at the effects of, in this case, allopurin. And we found that allopurin nicely relaxed the blood vessels, and you could block the effect with a KCNQ blocker, XE991. Allopurin is actually KCNQ5 selective. So if you look at the other neuronal channels, here's the Q5 is activated nicely, whereas the other ones are not, even if you go up to very high concentrations. So I'm not aware of any other KCNQ5 selective openers. Most of them also open Q4. So I believe this may be the first example, or there may be some examples that are not in the literature. We'd also looked at KCNQ1. There's a small effect of allopurin on Q1, but when we add Q1 with its native partners, such as E1 or E3, it goes away. So allopurin also is very good at activating Q5, but not Q1 or Q1E1. So it's quite KCNQ selective. So finally, my conclusions, neuronal KCNQs involved a neurotransmitter binding pocket, which has an affinity for GABA that's very similar to the tonic GABA levels in the brain. So we think that's a way for potassium channels in the KCNQ family to sense long-term changes, although we actually have a couple of mouse models now that we're using to test this, where we made a mutation in the binding site for GABA. I don't have data for those yet. The same binding pocket is exploited by medicines, but also by traditional medicines, plant-based compounds. And the final stuff I showed you was that KCNQ5 activation, very unexpectedly, is a unifying molecular mechanism for many different traditional botanical hypotensive medicines. I would like to thank my funding support, which has been obviously essential, my high throughput system, which is Rian Manville. Unfortunately, he's now starting his own lab, so he'll be his own high throughput system, but I have Katie to take over where he left off, and Tom Jeps in University of Copenhagen, and also Ben Katz, who's been great at figuring out which compounds are in the plants, so we can then go and find an actual compound, not just look at the plant extract. Okay, thanks very much for your attention.
Well, thank you, Jeff. That was very interesting. Yes, I can see a question over here. And then the second one, I think I saw a hand up over here somewhere. No,
thanks.
okay.
Over here.
Yep.
It's really beautiful work. I wonder, based on the last stuff about KCNQ5 in vascular smooth muscle and vasorelaxant or antihypertensive effects, did you ever look at heteromeric Q4, Q5, or combinations with the KCNE subunits to see if that had any impact on their responses?
Sure, so it's a really good question. So the question is, we looked at KCNQ5 activation. Did we look at KCNQ4, Q5 heteromers, which are also thought to be expressed in blood vessel smooth muscle, and also KCNEs, because it's thought that KCNE4 modulates Q4, Q5 heteromers. So we did. It's in the supplementary data, the reams of supplementary data. So Q4 reduces the efficacy. So our collaborator, Tom Jeps especially, is very interested in Q4, Q5 heteromers. We find that Q5 is much more sensitive to these types of small molecules. So whenever you put Q4 in there, there's still an effect, but it's not as dramatic. So the question is, I guess we don't know yet, how many of the channels in blood vessels are really Q5 homomers versus Q4, Q5? Maybe it's different for different blood vessels, which could be a really nice way of targeting specific blood vessels, because as we know, sometimes when you drop blood pressure in the core, you have dangerously high constriction in the brain and other effects. So it might be, we need to figure this out. It's a really good question, and we need to figure it out. Yeah.
I was curious about this model of the KCNQ3 sensing GABA, tonic GABA in particular. So, I mean, if you had tonic GABA, you'll think, more inhibition in general, and then you're activating KCNQ3, which potentially is even more inhibition. And is that, why would we want that, for example?
Sorry?
Why would you want that? Like, there's already tonic inhibition, and then you activate Q3 and inhibit potentially the neurons even more. Like, what kind of scenarios do you think that might be important?
So So the question is, why would you need to activate GABA, activate KCNQs when GABA already works on canonical GABA receptors? So KCNQ channels we found are actually more sensitive than most commonly expressed canonical GABA receptors. So this is a different, this is a different type of modulation, I think. That's one answer. The other answer is that these are expressed in different neurons. They're not always in the same neurons. Another answer is that KCNQs have been shown to modulate GABA release in itself, so there might be a feedback mechanism going on there. Another answer is that we have a paper that's just in review now where we've shown previously that KCNQ channels interact with sodium-coupled solute transporters. Now we know that that changes the way that GABA activates these channel complexes. And the final thing is that if you have an upregulation of other metabolites that are similar to GABA, you can completely change what's happening. So GABAA, which is an endogenous metabolite and possibly neurotransmitter, is very close to GABA. It's called GABAA, gamma amino hydroxybutyrate, I believe. That is a partial agonist of KCNQs. But it's much higher affinity. It's about 10-fold maybe higher affinity than GABA. So if that gets in there, GABA's not getting in. So it completely, I think that it's a way of sensing longer-term metabolic changes. The other thing we showed, which we published part of it, and we're going into more detail now, is that the ketone body, beta-hydroxybutyrate, which is the first ketone body that's produced upon going into ketosis, that also activates KCNQs in the same binding pocket. So when someone goes into ketosis, they can be activating their KCNQ channels in the brain via beta-hydroxybutyrate, which gets up to levels of maybe a millimolar in children on a ketogenic diet. The EC50 is about 1 micromolar. So we think that activation of KCNQs by beta-hydroxybutyrate could be part of the mechanism of the anti-epileptic effects of the ketogenic diet. So I guess my answer is the system's really complicated. There's so much going on with different metabolites and different protein complexes, different neurons in different locations, different timescales, different concentrations and concentration dependencies of the different channels. We're just at the very start of this. Like I said, we now have 2 mutant lines that we made where we've mutated the binding site for GABA in these channels, the tryptophan. So they work similarly at baseline, but they don't have that tryptophan. So it's what we want to do is say, is this important in physiology, or is it just something that we found that's really cool in vitro but doesn't do anything? I would be shocked if that was the case, but we will find out with these mice because you really can't argue if they have problems because they can't bind GABA on the KCNQ channel, then I think it's definitive. Yeah, so we just don't know at this point how important it is and why, I guess is the answer.
Yeah, so Jeff, I was just going to follow up on that question here.
Yes.
So GABA works on GABAA receptors and GABAB receptors, and it's released very, very, its release and where it gets to is very tightly regulated. And so you can imagine, you could easily imagine that you've got this site, but it's got no physiological significance. And I think of all the effects of GABA receptors that have been done over the years, I don't know if anybody's found some behavioral consequence that isn't explained by GABAA or GABAB. But I mean, it could play into it. But some of the things you're talking about with epilepsy and the ketogenic diet and things like that, I think they still tie into the regular GABAA receptor properties, right?
No, beta-hydroxybutyrate doesn't activate canonical GABA receptors.
No, sure. But then you're getting into, look, who knows? But
well, so at a synapse, GABA gets up to maybe 10 millimolar for a few milliseconds.
Yeah, tiny.
Okay, but tonically, we're talking about sub-micromolar. Sub-micromolar is, I think the standard I've seen is 150, 160 nanomolar.
Right.
So at that level, it's not as highly regulated. There's a tonic. GABA's in our bloodstreams as well. I mean, GABA's originally a metabolite. It's in animals and plants, I believe, way before it became.
But the GABAB receptors are regulating calcium channels and potassium channels, and that has a big effect on excitability already, right? So it's just, I mean, it's possible, right? But it just seems that somebody would have held their hands up and said that there's something that we haven't nailed yet about the GABAergic system, right?
Yeah, I just don't think people are looking along the timescales that one would be talking about. The other thing.
And the M current, is tied into muscarinic regulation.
Yeah, well,
right?
you know, so yeah, that's, if you look in vivo, a lot of the effects in, a lot of, the effects of muscarinic inhibition on the M current are actually very difficult to see in a lot of neuronal populations. So what we show is that that GABA completely overrides the effects of muscarinic inhibition. So when we remove, when we stimulate muscarinic receptors to inhibit KCNQs, and then we add GABA, GABA overrides that completely. So it can activate even in the absence of PIP2.
Because it's like some sort of allosteric modulator, in fact.
Yeah, it binds to that well, we now have evidence, we didn't publish it there, but GABA binds to the pore and changes the conformation of the pore. Basically, we know that because it changes the permeability, it changes the selectivity. So you know, it's one of these things where we looked at it and we did every control under the sun. Just because someone hasn't seen it before doesn't mean it's not happening.
So we're going to take one last question from Amy.
Yeah, just quickly. So I'm wondering if in the context of a native brain slice or tissue, if you inhibited the breakdown of GABA that's been synaptically released, like with vigabatrin or something like that, do you see a change in the membrane potential that could be attributed to KCNQ channels?
You'd have to. So we did very minimal recordings in neurons, because that's not really our thing, although we're transitioning now and building brain slice rigs so we can do that stuff. But we showed in neurons that we see the same, we basically see activation of M current with GABA. That's as far as we've got. I've talked to some people who are very excited about this finding and said, we think that in a certain type of neuron, this actually could be important. I won't go into the details. I don't want to steal their line of experimentation. But they're saying that what we're seeing could explain, in a specific neuronal type, the effects of GABA that aren't explained by canonical GABA receptors. Yeah, but and this is a really cool in vitro finding right now, and in the future, I'm hoping we'll figure out the physiological relevance.
Great, well, thank you. And I think it is a really interesting example of kind of reverse discovery, going from an exogenous compound through to discovering the endogenous molecule. So thank you.
Thank you.